Good morning, everyone. Welcome back to our last lecture with the content. We will meet on Monday, but it's going to be a question and answer kind of session. Ensley is finishing up the review for the final exam. So over the weekend, please be sure to take a look at the review at the study guide and compose your questions and come on Monday and ask them, okay? Today's lecture is going to be on biotechnology. Are there any questions before I get started? Okay, see a question in chat. For extra credit, do we answer the questions in bullet points or a paragraph? You can answer the questions in bullet points. Ideally, like you will have a sentence for each answer, and you can combine it in a paragraph. But if just answering the questions in the bullet points makes it easier for you, just go ahead and do that. Any other questions? Okay. So biotechnology. So DNA technology has become very widespread and it's literally changing the world. DNA-based technologies have affected nearly every imaginable area in our society, and it's a powerful tool in research, in medicine, in agriculture. We eat a lot of stuff that is genetically engineered, in criminal justice, and in many other in many other fields. So scientists can manipulate DNA in many ways. They can change its sequence, copy it, switch the genes on and off, identify pieces of DNA that are of interest and transfer it between species, such as, let's say, the gene that, for instance, is toxic to insects. The gene that contains a copy that is contains a recipe for a protein that is toxic to insects. Well, it can be transferred to a plant, thus eliminating the need for using herbicides. Because if an insect eats a transgenic plant with the gene transplanted to a plant that is toxic to to an insect, the insects will start dying by themselves once they eat the plant. So manipulating the DNA for practical purposes is called DNA technology. And changing the DNA changes the proteins that cells can produce, because remember, the DNA, it contains the recipe for the proteins. So if the DNA has changed, the proteins that are expressed are changed as well. DNA can be manipulated in tubes, in living cells, and in entire organisms. And reading the sequence of DNA is used to identify individuals, diagnose medical conditions, as well as study evolution, as we've seen before. So transgenic organisms, they contain DNA from multiple sources. Since all of the species use the same genetic code, the cells can express genes from any species. So if we take a gene from one species and somehow transfer it to the cells from another species, the gene will still be expressed because the proteins, they will still synthesize RNA, messenger RNA. This messenger RNA will carry this recipe to a ribosome. And within the ribosome, tRNA will still bring in amino acids to create a protein. So a transgenic organism is an individual that receives a so-called recombinant DNA, or a genetic material that scientists have spliced together from multiple sources. So often the, you know what, I'm not going to touch it, just scratch it out. I'm not going to go there. So transgenic bacteria have many uses. The bacteria engineered to produce the desired molecules, especially drugs. So insulin is a prominent example of how transgenic bacteria can be used in medicine. The bacteria have also been engineered to produce blood clotting factors that are useful for people with blood disorders, as well as fertility hormones. So others are engineered to degrade petroleum, pesticides, and other soil pollutants. So bacteria can be our friend and do a lot of useful things if we change their genetic code a little bit to make it useful for us. So how is a transgenic organism created? What is going on there? So scientists use plasmids, which are a small circle, which is a small circle of double-stranded DNA. This is a plasmid here that is separate from chromosomes. So we have DNA in chromosomes, but there's also some circled DNA that floats around that is not on that is separate from chromosome, that does not form a chromosome. So plasmids can replicate themselves inside a host cell, and since it's a DNA strand, and be used for expressing the genes they carry. So next, I'm going to go over for a little bit how these recombinant plasmids are formed. So to begin making transgenic organisms, scientists combine a plasmid with source DNA, the DNA they want the transgenic organism to have. So this is step one. Okay, look at the DNA. For instance, we want a bacteria to produce insulin. Bacteria naturally do not produce insulin, but we want to genetically engineer a bacteria to start doing that. So we first look at the human DNA and locate the gene that contains the recipe for insulin. Now let's say we found this gene, and then we can isolate it. Okay, we can isolate this gene. Then we take the plasmid or the circular DNA, and we splice it. We kind of cut it open and insert the gene into the gene for production of insulin into the circular plasmid. And as a result, we have recombinant plasmid that contains some of the original DNA and some new DNA, as a new gene, I should say, for production of insulin. So this plasmid then needs to be inserted into the bacterial cell somehow in order for the cell to start making insulin. So how is that done? One way to put plasmid into the bacterial cell would be zapping the cell with electricity. When the bacterial cells are zapped with electricity, the channels within the membrane form. So within the membrane, some openings are forming through which the recombinant plasmid can enter into the cells. There are also so-called gene guns that can shoot DNA directly into a cell. And if we're not working with bacteria, but if we want to inject the recombinant plasmid into an animal cell, for instance, usually a virus is used. So a virus infects the recipient cells. Well, for bacteria, technically, it also can be used, but using viruses to transfer the recombinant plasmid into an animal cell is very common. So scientists use bacteria to create transgenic plants. The bacteria that is used is called agrobacterium. So if the agrobacterium is not genetically manipulated, well, let's say a natural agrobacterium, the way it works, it's when it infects plants, it injects its own DNA into plant's nucleus, and not plant's nucleus, the nucleus of the cells within a plant. And it starts, as a result, plants start forming abnormal growths on their bodies. Okay, so this is like the natural way that agrobacterium works. And the plasmid that causes this abnormal growth on plants is called TI plasmid. And TI stands for tumor-inducing plasmid. So if we genetically engineer, we can genetically engineer this TI plasmid, change its, like insert the gene of interest into it, the gene with the recipe for a particular protein. For instance, a protein that helps the plants to be resistant to herbicides. then the agrobacterium will help us deliver this TI plasmid into the plant cells. So the agrobacterium infects the plants, but instead of growing tumors, the plant starts being resistant to herbicides. So we edit out the genes that induce tumors and instead include the gene, for instance, for herbicide resistance. So this herbicide resistance is very useful in agriculture because when you have a crop of interest, and it usually is, some weeds are present as well. It's not usually like monoculture crop if it's not treated. So farmers are interested in getting rid of weeds and leaving only the culture of interest because to increase the yield per unit area. And if they kill the weeds, if they decide to kill the weeds with herbicides, the non-genetically engineered crop would die as well because the herbicide does not distinguish between crop and weed. But if the crop is genetically engineered to be resistant to herbicide, then only the weeds will die, and the crop culture of interest will survive and thrive. Okay, so uh, to infect plant cells with, or to genetically engineer plant cells, scientists usually use agrobacterium, which naturally injects their recombinant TI plasmids into the plant's nucleus. So plasmids express new genes in host cells. So once the plasmid enters the host cell, the cell can express genes carried in the plasmid, like herbicide resistance. And this plant expresses bacterial gene for herbicide resistance, and this fish expresses a jellyfish gene for glowing color. Okay, so fish do not naturally glow. Okay, so this fish was genetically engineered with a gene from a jellyfish to glow in the dark. So the beauty of the genetic engineering is that once you infect one plant cell with agrobacterium, I mean, once the DNA within one plant cell is changed, when this plant cell divides, the daughter cells will also have this new gene for herbicide resistance. So you only need to infect the plant once. The rest of the cells will just, once the cells divide, the daughter cells will have the same DNA. So for animals, scientists do not use agrobacterium. They usually use viruses. And so genetically modified viruses carrying desired genes instead of the viral genes can be used to infect early embryonic cells. When cells divide by mitosis, as the animal grows and develops, they pass along recombinant DNA to all the cells in the animal. So remember, the viruses are just genetic material in a protein envelope. And if this genetic material is not harmful, if this genetic material is something of interest to us, like if we ourselves, decide what this genetic material has to be, the way the virus will infect the cells will remain the same. But instead of producing something harmful to the body, it will just inject the DNA into the cells that will express some useful kind of protein or enzyme. But remember, viruses, they kind of infect cells, and this is useful for transfer of modified DNA into the animal cells. An useful aspect of biotechnology is the technology called PCR, or polymerase chain reaction. This is widely used technology in medicine to diagnose certain conditions, particularly infections. Because PCR, well, what PCR is, or what it's rather useful for, it's used to replicate a small amount of DNA. So if we have very small amount of genetic material and not enough really to analyze, we can take this small amount of genetic material and throw it into this vessel, which will contain DNA polymerase, which is an enzyme used to replicate the DNA, a bunch of nucleotides and a bunch of primers. So Once we throw in the little piece of DNA, it will be replicated over and over and over and over and over again. So it's basically a copy machine. Okay, PCR, think of it as a copy machine for DNA. And eventually there will be enough DNA to conduct multiple analysis with and do multiple sequencing. So as I mentioned, this is often used in diagnosis of infections. It also is used in the criminal justice system. And in fact, it's been so useful that over time, as this PCR technology has become widely available, the DNA exonerations in the U.S. have risen substantially. Okay, so DNA profiling basically strengthens the criminal justice system, and due to DNA profiling and thanks to PCR in particular, the number of innocent and wrongfully convicted people being exonerated and released from prisons has increased. So before 90s, it was very tough to collect the DNA and actually do anything with it. But because the amount of DNA was small, it was often degraded. But with the PCR technology, this DNA can be replicated over and over and over again, and the scarcity of DNA does not become an issue anymore. So the stem cells and cloning are another exciting aspects of the biotechnology, and they add new ways to copy cells and organisms. Stem cell research often sparks public debate because of the origin of stem cells, which are from embryos. And some people see it as a useful technology, while others see it as unethical. And both arguments of both sides are very valid. So the discourse over stem cells and cloning combines science, philosophy, religion, and politics. So the pros of using, like one of the pro arguments of using stem cells is basically the stem cells from embryos can be turned into anything, into any cell within our body. So it can, if the liver is shutting down, then the stem cells can be turned into liver cells and kind of regenerate the organ. If the kidneys are shutting down, the stem cells can be used to grow, like regenerate the kidneys, and so on. However, because the stem cells that can do that come from embryos, the hard decision becomes, where do you get these embryos, and how ethical is it to obtain them, and how ethical is it to create life just to destroy it for something else. So stem cells are the kind of cells that are undifferentiated. Animal development relies on stem cells. The very first cells that form in an embryo are stem cells. So note that this blob of cells will later on differentiate into skin, into muscle, into neurons, into blood cells, into bones, into anything. But initially, the embryo is just a blob of cells, a blob of stem cells, to be more precise. And so these stem cells are special because they are unspecialized. They retain the ability to differentiate into different type of cells. The adults also have stem cells. However, they cannot differentiate into many organs. So the stem cells within our body are typically contained in the bone marrow. However, these stem cells can only become red and white blood cells, fat and bone cells. So these stem cells can kind of regenerate whatever is around them, so since red and white blood cells are formed in the bone marrow, the stem cells within the bone marrow can, generate the red and white blood cells. So adult stem cells, although useful for regenerating these three type of cells, blood, fat, and bone, they cannot be turned into kidney cells. They cannot be turned into liver cells. That is why scientists keep turning back to embryonic stem cells, and we keep having ethical issues about using embryos to do research. So cloning creates identical copies of an organism. Cloning or asexual reproduction is very common in nature. among single-celled organisms such as bacteria, archaea, and many protists, as well as among plants. I mean, the plants are the cloning among plants is very widespread in nature. Just think about like when you take a piece of plant, like when you take a leaf from a plant and stick it into water, and it will grow roots after a couple of weeks, and then you can take this leaf and plant it into the ground, and the whole new plant will grow from that leaf and little bitty roots. And this is cloning because the plant that you will grow, it's going to be genetically identical to the origin plant, to the plant that you got the leaf from. So uh, plants can be cloned easily, right? However, with animals, it's not the case because animals, they do not reproduce asexually; they produce sexually. So cloning in animals is done using somatic cell nuclear transfer, and I'm going to go over it in a little go over it in a more detail a bit in a bit. So the first clone animal was Dolly the sheep, and the way it was formed, the way it was created was that the nucleus from an adult sheep cell was used to create Dolly, the clone sheep, whose DNA was identical to the donor. So what is this somatic nuclear transfer? So a diploid nucleus, a nucleus with the two copies of the chromosome from an adult donor cell or from an adult sheep, is placed into an egg cell of a donor. Okay, so because the this nucleus from the diploid nucleus is placed to a donor egg cell, no fertilization is needed, and this donor egg cell with implanted nucleus, which contains all the genetic information, starts dividing and forming like a zygote. I'm sorry. First, the zygote is formed, and then the zygote starts dividing and developing into a new organism. So before that is done, though, this egg has to be implanted into a surrogate mother animal and goes through normal mitosis and development. And when the new animal is born, it is a clone of the adult donor animal. So again, uh, first we identify like the sheep that, well, the way that Dolly was created. I'll go to the concrete example, a specific example. The way that Dolly was created was the mother, quote unquote, sheep cell was kind of the nucleus from the cell was extracted and placed into a donor egg cell with kind of no genetic material. And this egg cell was implanted into a surrogate mother and developed into a baby sheep. Okay, so the egg cell contained only the genetic material from Dolly's quote unquote mother. Well, Dolly did have a mother because she was cloned. So yeah, this is what nuclear transfer is. So there are lots of issues around ethical issues around the question of human cloning. It's on one hand, it could help human cloning could help infertile parents have children, and could be used to harvest and human cloning could also be used to harvest embryonic stem cells. But many ethical issues surround cloning of our own species. So this part is would be very useful for how do I put it, personalized medicine. For instance, let's say you have kidney failure and you need a new kidney, and you're looking for a donor. So instead of looking for a donor, like you could have yourself cloned, and create like the embryo of yourself could be created, and the embryo would not let you would not let the embryo develop into a full adult or even a baby. You would harvest the stem cells from the embryo. You would eventually essentially kill it and harvest the stem cells to regenerate your own kidneys. Okay, so this is kind of the big idea behind the pro for human cloning. So you would not completely clone yourself. You would just clone yourself for the embryonic stem cells, which could be used to regenerate your own organs. Um, However, again, this embryo would be could develop into a human being, and killing the embryo is considered unethical, but by many people. Mm, all right. So another useful example for biotechnology is so-called PGD, or PGD stands for pre-implantation genetic diagnosis, and it's used to help reduce the odds of having a child with genetic disease. And so, first, a man's sperm fertilizes several of the woman's eggs in vitro, meaning in a petri dish, and then as one cell from the embryo is taken away for analysis for this pre-implantation genetic diagnosis. And if some genetic defects are revealed using this analysis, then this embryo is not implanted into a woman's uterus. So usually, like with if people have fertility issues, they can resort to in vitro fertilization, meaning the multiple embryos are created in a petri dish from women's egg cells and men's sperm, and these embryo to decide which embryo to implant to reduce the chances of having a child with a genetic disease. One cell from each embryo can be harvested and analyzed for genetic defects, and and the most healthy embryos then are implanted into the woman's uterus. So another example of biotechnology is gene therapy, and gene therapy uses DNA to treat disease. And it may someday provide new treatment options for genetic diseases. So the overall goal is to supplement a faulty gene with a normal, healthy version. So gene therapy could be used for a disease called cystic fibrosis, which we've gone over, I believe, in some past lecture. So in cystic fibrosis, it affects the lungs, and it causes the lungs to have this thick mucus that kind of prevents the person from breathing very efficiently. And cystic fibrosis is caused by a mutation in a gene on chromosomes on the seventh chromosome. So the result is some abnormal proteins in the lung cells that impair the lung function. Researchers start with a virus that can infect the lung cells. Okay, and they remove the viral DNA and make the recombinant viruses containing healthy versions of this so-called CFTR gene that that contains the mutation if the person has cystic fibrosis. So then the patient inhales the recombinant viruses, and recombinant viruses or genetically modified viruses, they inject their genetic material, including a healthy gene, into the lung cells. Then lung cells can produce normal CFTR proteins that will not cause this thick mucus buildup. So this is an example of how gene therapy can be used in the medicine and kind of treat the genetic conditions. And finally, CRISPR. There was a lot of news about CRISPR a couple of years ago, and it has been presented as a revolutionary technology for gene editing, which it is. So CRISPR technology, it uses guide RNA to find the gene that they want to edit. So the steps in gene editing using CRISPR, first, the target sequence of DNA to be modified is determined. Then the guide RNA is engineered, and this guide RNA is then kind of injected into the nucleus, and this guide RNA binds to a target DNA sequence. Then the enzyme called Cas9 follows the guide RNA to the target sequence and cuts both DNA strands. So it kind of envelopes the target sequence and kind of cuts the DNA over here or over here. So then depending on the researcher's goal, the cut DNA may be turned off, or it can be modified with the added nucleotides. So we can identify a gene, engineer an RNA that would bind to that gene, then inject the enzyme, which would bind to the target gene, which would locate this RNA and bind to the locus on the DNA, and then do whatever with it. The gene can be turned off or edited out of the DNA, or it can be modified to do something else. So this technique allows researchers to quickly and easily modify one specific gene in an organism without affecting any other part of the genome. And that's why it was so revolutionary. All right, that's all I have for today. Do you guys have any questions?
So are the glowfish born with the glow ability, or do they use like a gene gun to like inject it?
Viruses. So the glowfish are not, well, they can be born that way from the parents that are glowing, but the original glowfish was produced using viruses. So the viral genetic material was removed from a virus, and the target gene for glowing from the jellyfish was injected into the virus. Then the fish was infected with this virus. And remember, the way viruses work, they find the cell and infect it, meaning they inject their genetic material into the cell. So once one fish was infected, or maybe two fish were infected, the baby fish that will result from those two fishes will also have the same genetic material as their parents, meaning they will have they will glow in the dark.
That's cool. I was talking to someone before and they said that they made a cat glow.
Huh,
Have you
interesting.
heard of that?
No, I have not heard of that.
I think they use like almost the same technique with the fish, but they did it on cats.
Yeah, it is possible. I mean, researchers made the mice glow, so I guess that the cat could be made to glow as well. I mean, why would you want that? So you can see it in the dark.
It's like an antique animal. Um, what was the third way that genes can be changed? It was zapping with cells, gene guns, and what else?
Viruses.
Okay. Alrighty. I think that's it for me, but thank you.
Yep. Anyone else? Well, okay then. So we'll see each other on Monday. Keep an eye out. Ansley will post a study guide today, and keep an eye out for it. Go over it today or before lecture on Monday, and bring your questions to Monday's lecture. And you have a good weekend, guys. Almost done with the class, okay? Last push.